Pakistan, especially for the northern areas, and we are improving some of these traits with the gene editing. So, so when we are talking about the genotyping platform, each genotyping platform which has a strong application, it should have two properties. First is the flexibility, because in the breeding program we are dealing with very large number of populations. So the genotyping platform should have the ability to genotype any number of individual varieties with any markers. And the second one is the cost effectiveness, because cost is the major limiting factor in the breeding program. So based upon these considerations, we have developed a genotyping platform using the functional markers, because these markers have very broad breeding application, and these markers are very cost effective. And with the help of these markers, we practice not only the marker assisted selection, but we can also practice the genomic selection. So because new genes they are being discovered with a rapid pace, and new technologies they are being introduced, so we should continuously update the market, and we also continuously update the genotyping platforms. So based upon these considerations that a marker platform should be a flexible and cost effectiveness, we developed three different genotyping platforms. First is the Cas marker platform. So these are the uniplex markers, and they have a greater application, and they are very high throughput markers. So here you can get an idea that if we are using the conventional genotyping technique, we can only get 0.1 million data points in 1 year. But if we are using the Cas markers, we can reach up to 36.8 million data points in a year. And these markers are also very cost effective. And we developed this marker platform almost 7 or 8 years ago, and it was rapidly adopted by many breeding and genetics programs. And according to our estimate now, more than 40 genetics and breeding programs all over the world, they are using our Cas markers. So as you know that this is the list of Cas markers which we have developed, and the varieties which we have screened with the help of these Cas markers. But these Cas markers are not sufficient, because these are the uniplex markers, and the challenge we have ahead, we have to improve the yield and the quality of the varieties. So these are all controlled by the quantitative traits, and the good strategy to deal breeding with the quantitative traits is the genomic selection. And for the genomic selection, we need high density markers. So develop more high density marker platforms. We are now focusing on the genotyping by targeted sequencing. So it is a new approach. It is NGS, next generation sequencing based approach, and it is much more cost effective than the SNP chips. So all our efforts in the future, they are based upon the genotyping by targeted sequencing technology. So in the beginning, almost 3 years ago, we developed these two platforms. So first is the GBTS 200. This is a platform for 200 markers, and include all the functional genes plus some markers for the diversity analysis. And the cost of this platform was very low. The second high density platform which we developed was the GBTS 40K. So it has 40,000 markers, and it could be a very good marker platform gene discovery, for the genomic selection, and for the gene characterization, because we included all the functional genes in GBTS 40K. But again, the cost of GBTS 40K was very high, and we cannot use this platform for the breeding application, specifically for the genomic selection, because according to our estimate, an ideal platform with the breeding application should have the cost of less than $10 per sample, because that is the phenotyping cost of one yield plot at most of the breeding programs. So if we can replace this phenotyping cost with the genotyping platform, that could be the ideal situation. So that's why for this purpose, we are trying to develop a very showcase platform by the help of GBTS technology, and it is a 5K GBTS platform. So it would have 5,000 markers. It will include all the genes and the QTLs which are being used in the wheat for the wheat breeding, and plus it will have a very high coverage of the SNP markers from every genetic region of the wheat. So that could be the ideal platform for the breeding application, and we could use this platform for the genomic selection. And the good thing is that the cost of this platform is almost $8 per sample. So this is an overview of this platform. We have functional genes at the top, then we have all the reported QTLs, we have genome-wide SNPs, and we have some SNPs from the whole genome resequencing data. And the beauty of the GPTS platform is that we can easily update these platforms time to time. While if we are using SNP chips, it is very difficult to update the SNP markers on the SNP chip. So that was all about the genotyping platforms which we are developing. The second part of the presentation is about the introduction of the facultative wheat to Pakistan. Currently, there is not a single facultative wheat introduced to Pakistan. So we are trying this effort because our northern areas, they are quite suitable for the facultative wheat. So we introduced some of the facultative wheats to Pakistan, and we did the field trials. You can see all in all the northern areas, and we are doing these field trials since last 4 years. And what we have observed that these two of these facultative wheat lines, they have almost 7 to 10% yield advantage compared to the local check. Because currently all the varieties usually from the NARC, they are being cultivated in the northern areas. So we hope that if we are able to introduce the facultative wheat for this region, we can have some very good yield advantages. So these are the two lines which we are trying to introduce to the northern areas. This is the field trial at NARC. The third part of the presentation, that how we can use these new breeding technologies like the genome editing to improve specific traits. So over the years, the collaboration between CIMIT and Harvest Plus, it was very beneficial, and they have introduced almost 5 different high zinc varieties, including the first variety was Zincol, and second is Akbar and Nawab, and recently Tarnab has introduced 2 more high zinc varieties. But unfortunately, there is not a single high iron variety in Pakistan. And iron and zinc, they are very important from the nutritional point of view, and they are very important for the good health and well-being. And if we can fortify wheat varieties with high iron and zinc, we can also give a very good health benefits to the people, because we have a very high consumption rate of wheat in Pakistan. So the first part was that how, because Zincol was the first variety released in 2016, but we still don't know that what are the major genes in the Zincol variety which are involved in zinc homeostasis. So we did RNA-6. We identified all the differentially expressed genes in the developing grain of Zincol, and then we identified that what are these genes, what are their functions using the transcriptome approach, and then we were able to identify almost 200,000 SLPs and 17,000 indels specifically in the developing grain of the Zincol variety. So this number was very large. So what we did in the next stage, that we pinpointed that what are the key genes which are in the Zincol 16 variety, because we had these SNP data from the very large Chinese population, natural population, for which we have the iron and zinc data. So we did some association analysis, and by this association analysis, we identified that ZIP4B gene. It was very differentially expressed in the grain of Zincol, and the Zincol type allele of the ZIP4B gene, it was associated with the high zinc contents, and this association we have observed from more than 6 environments. 6 environment zinc concentration data from a very large population. So second gene which we identified is the NAD-glucodeamidase aminotransferase gene. It is also a zinc or metal homeostasis gene, and we identified that Zincol type allele is consistently increasing the grain zinc contents in wheat, and we have developed a marker, so we can also introduce this gene with the help of marker-assisted selection. So this transcriptome based approach was very beneficial, that how we can pinpoint the genes which are associated with our desired phenotype. So iron homeostasis, as I told you that there is no single high iron variety in Pakistan. So we are trying efforts that what could be the key regulator of the iron homeostasis and how we can improve the iron contents in these. So gene editing is a very promising strategy. What we did that we identified a master regulator which is a VHLS transcription factor, and we know that this master regulator is a negative regulator. So if we are able to silence this master regulator, which we usually call Brutus gene in the Arabidopsis and HRZ gene in the cereals, so it is a negative regulator. So our approach was that how we can silent or knock out this gene with the help of CRISPR-Cas, so we can have more iron contents in the grain. And the good thing is that it is a ubiquitous gene, means it is also involved not only in the iron acquisition, but it is also involved in the iron transport and iron storage in the grain. So we silence this gene with the help of CRISPR-Cas, and you can see the results that compared to the null mutant and the control variety, both we have high iron contents in the leaf and the grains of the zinc, and we did this gene editing in the zincol cultivar. So our idea is that because zincol also already has high zinc content, and if we are able to increase iron contents, that could be a very good ready-made product for the nutritional point of view. So you can see that both leaves and the grains, they have significantly high irons by silencing or by gene editing the HRZ gene. And if you look at the iron and zinc data with the ICP-OES technology, you can see that this black dotted line is the iron content in the null mutant, and the gray dotted line above is the target set by Harvest Plus. So all these gene edited lines, they have more high iron and zinc contents more than the target set by Harvest Plus. So we because these results were very promising, so UK started a new center which is the UK CGIR center, and by the help of this center, in the future we will be evaluating these lines in Pakistan, Kenya and Egypt. So we can also commercialize these lines if the regulations allow these gene editing technologies, products of these gene editing technologies. So thank you. That's all about my presentation.
Thank you, sir, for your nice presentation. Due to shortage of time, I would like to take one question if anybody has.
Just thanks for the presentation. What was the principle of these 50,000, 5,000 SNPs and 40,000 SNPs? Is it based on cellular hybridization?
No, these are the seeds from the whole genome sequencing.
Yeah, you have markers attached to the chips, right?
Yes.
But how do they identify, the different genotypes or different rice, wheat lines?
No, it is not based upon hybridization. It is a sequencing technology.
Is amplisync?
Yes.
Amplisync? Does it apply amplisync?
Yeah.
Okay.
It is ampli sequencing.
Okay, I got it.
We have target. We enrich that target with the PCR markers.
Yeah, I got it. The other question is about, was there any collateral damage after you did editing to the zinco with respect to the ferrous homeostasis?
We identified because we had almost 50 gene edited lines, and some of these lines couldn't survive because if we have more iron in the leaves and in the system, it could lead to the toxicity. So we have several lines which had a very high iron toxicity and they couldn't survive.
These were selected.
So these are the lines which could survive this high iron.
Thank you.
Dr. Saab, I have just one short question. Is there any limit of this zinc iron you would be expecting that this will be normal when going to gene expression? तो कितने लेवल तक अमाउंट जो सेंसाइज करे जो बॉडी के लिए मतलब टॉक्सिक ना हो?
Harvest Plus, they have already set the targets, which is almost 50 ppm or 50 mg per kg. So this is the target set by Harvest Plus. So each grain, each variety should have almost 50 ppm of the iron and zinc.
Thank you, Dr. Ahmed Saab. Thank you very much. Our next speaker for this technical session 3 is Dr. Zahid Mahmood, who is an eminent scientist of NARC, National Agriculture Research Council. He is the pioneer in speed breeding in Pakistan.